Why are PCR machines, or polymerase chain reaction machines, considered indispensable in the realm of scientific research? The answer lies in their exceptional ability to amplify minimal amounts of DNA, enabling scientists to study genetic material in depth. But how does this remarkable machine work, you might ask? Well, it's time to find out. Imagine you're a detective, armed with a magnifying glass, searching for a single specific footprint in a city bustling with millions. That's what it's like for scientists trying to study a single bit of DNA. Now imagine if you could take that single footprint and clone it 1,000,000 times over, making it much easier to find and study. That's what a PCR machine does with DNA. The PCR machine operates in 3 main stages: denaturation, annealing, and extension. In the denaturation stage, the DNA sample is heated to around 95 degrees Celsius. This heat breaks the weak hydrogen bonds holding the 2 strands of the DNA helix together, causing them to separate. Think of it as unzipping a zipper. Next comes the annealing stage. At this stage, the temperature is lowered to about 55 degrees Celsius. This allows short pieces of DNA called primers to bind or anneal to the separated DNA strands. These primers act as starting points for a new DNA strand. It's akin to marking the right spot on a treasure map. The final stage is the extension stage. Here the temperature is raised to around 72 degrees Celsius, the optimal temperature for an enzyme called Taq polymarase. This enzyme moves along the DNA strand, adding nucleotides to extend the new DNA strand from the primer. It's like a builder laying bricks to construct a wall. These 3 stages are repeated around 30 to 40 times in a cycle, each time doubling the amount of DNA. So from a single piece of DNA, you can generate over 1 billion copies. It's like starting with 1 tree and ending with a whole forest. So, why are PCR machines so important? Well, they enable scientists to study minute amounts of DNA in great detail. They're used in a wide range of applications, from forensic science, where they can help solve crimes by identifying suspects from tiny DNA samples, to medical research, where they can detect diseases and genetic disorders. In the field of evolutionary biology, they allow scientists to study the DNA of extinct species. In the fight against pandemics, they have an essential role in testing millions of samples to identify the presence of a virus. In summary, PCR machines are the unsung heroes of the scientific world. They take what is invisible to the naked eye and make it not just visible but plentiful enough to study in depth. From crime scene investigations to diagnosing diseases and understanding our biological past, these incredible machines are an essential tool in our quest to understand the world around us. So, the next time you hear about a breakthrough in science, remember the humble PCR machine. It's probably had a hand in it.